Chapter 26 is looking at phylogeny and the tree of life. Definition of phylogeny is the evolutionary history of a species or group of related species. So the example that they talk about here is that a phylogeny shows that legless lizards and snakes evolved from different lineages of legged lizards. Systematics is defined as the part of science, the discipline of science, that classifies organisms and determines their evolutionary relationships. So we're going to spend a lot of time with systematics in Bio 7. We already know that Carl Linnaeus was responsible for devising the system of taxonomy, and he's infamous for his two-part naming of species, that binomial nomenclature, but he's also responsible for producing hierarchical classification as well. We've already talked about in our binomial nomenclature, the first part of the name is the genus. The second part we call the species or the specific epithet, which is a description. So the first letter of the genus is always capitalized. The entire species name is italicized or underlined if you're writing it. Both parts together name the species. You can't just have that specific epithet by itself. Linnaeus was responsible for producing that grouping that you may have heard of before. So what we recognize in essence today is domain, kingdom, phylum, class, order, family, genus, and species, going from very broad to very narrow. So genus and species together, you're looking at a single population of organisms. You need to know this for both lecture and lab. Taxon is defined at any taxonomic unit at any level of the hierarchy. They make the statement here that the broader taxa are not comparable between lineages. So looking at an order of snails isn't equivalent to looking at an order of mammals. Characteristics are going to be different. The genetic makeup, totally different. So here, this kind of gives you an idea of going from very broad down here at the bottom to very narrow at the top, looking at an individual species. Okay, so this particular organism comes from the domain eukarya because it's made up of eukaryotic cells. It's found in the kingdom Animalia, and if you look here, there's several different kingdoms that can exist. Phylum Chordata, which comes from that kingdom Animalia. Sorry, I'm pointing out the wrong thing here. Here's domain eukarya. These are the several different kingdoms. These circles that you're seeing here are the different phyla that can exist. One of those classes is Mammalia. Of the mammals, there's an order that's called Carnivora. Of the order Carnivora, there's a family called Felidae. And within that family of Felidae, there's a genus, Panthera, of which one individual species is Panthera pardus, which is our leopard. The evolutionary history of a group of organisms is represented by that branching phylogenetic tree. And we're going to look at several different trees in this chapter. This gives you an idea of basically what we just looked at, starting at order. So here's our order Carnivora, of which our Panthera pardus is a species, but also the Taxidia, Taxus, Lutra, the Canis latrans and the Canis lupus are also species that are found within the order Carnivora. We can take this order Carnivora, divide it up into 3 different families by the organisms that are represented here: Felidae, Mustelidae, and Canidae. And Felidae are cats, of which Pantheropartus is one. Mustelidae is talking about organisms that have a very musk-like odor to them. Taxidia and Lutra are both species within this particular family. And a close relative would be the Canidae, which would be the genus Canis, and this brings us to the Canis latrans and the Canis lupus, which are both basically cousins of each other. Same genus, different species. Linnaean classification and phylogeny can actually be a little bit different from each other. So systematists have proposed a classification system that is trying to just group its common ancestor and all of its descendants that could come from that common ancestor. So let's describe a phylogenetic tree. So phylogenetic trees looking at evolutionary relationships. Each branch point is representing the diversion or the branching off, if you will, of 2 evolutionary lineages from a common ancestor. Sister taxa are groups that share an immediate common ancestor that is not shared by any other group. Okay, so you should know what a branch point is. You should know what sister taxa are. This kind of gives you an idea of the parts of a tree. So common ancestors back here. This is showing you an evolutionary lineage. This would be the other lineage to include all of these organisms. Okay, here as we're looking down the frog lineage, here could be an ancestor in the frog's evolutionary lineage. This one down here is showing sister taxa because these guys have diverged from this common ancestor. Okay, these guys aren't represented as sister taxa at all. Phylogenetic trees can really be drawn any way that you want to. They can be horizontal, they can be vertical, they can be diagonal. The shape of the tree doesn't necessarily change the relationships that are being shown. So this kind of shows you a vertical tree, a diagonal tree that can be drawn from the same information that we saw previously. Tree branches can be rotated around a branch point, and that rotation is not going to change evolutionary relationships. Keep that in mind. Okay, so here we have rotation that could occur around this branch point, around this branch point, or possibly around this one. So here we've changed our order. Our frogs are up here. Human and chimps ended up here. Lizards here. And fishes ended up down here. Okay, so doing rotation in all 3 of these places gives us another way to look at things, but the evolutionary relationships here haven't changed. You're just looking at them in a slightly different order. A rooted tree is going to include a branch that represents the most recent common ancestor of all the taxa in the tree. So a rooted tree, think of it as having a nice solid base. Okay. A basal taxon is that species that's going to diverge very early in the history of the group that you're looking at. And we're going to see that this basal taxon actually will originate near that common ancestor of the group. So it's going to go way back. This kind of gives you some of the points that we've been talking about. Here's our most recent common ancestor. Right there's a branch point. These two are considered sister taxa. These two would be considered sister taxa as well. Taxon F is our basal taxon because this is the one that is most like what our common ancestor was back here. Phylogenetic trees are going to show patterns of descent. Okay, so not necessarily phenotypic similarity. So the species that are next to each other may not necessarily look like each other. Phylogenetic trees do not indicate when species evolved, nor do they tell us how much change has occurred in that lineage. You should never also assume that a taxon evolved from the taxon next to it. Okay, so be careful with that as well. To help infer phylogenies, systematists are going to gather information about morphologies, so that's what the organisms look like. They'll gather information about their genes, so their genetic makeup, and also the biochemistry of their living organisms, how the enzymes and chemicals all work together within that organism to do what they do. Phenotypic and genetic similarities due to shared ancestry are called homologies. Now that word homology is very important because we're talking about things that look alike, they act alike, but there's also a component of shared ancestry. Okay, they have a recent common ancestor at some point in their makeup. Okay, so in terms of morphological and molecular, looking at the genetic information, this is tied into the phenotypic and genotypic, if you will, similarities. So these are our homologies. Organisms with similar morphologies or DNA sequences, so those that have a lot of homologies are going to be more closely related to each other than organisms that either have different structures or different DNA sequences. Okay, so keep that in mind. Homology is similarity due to shared ancestry, common ancestor. Analogy is similarity due to convergent evolution. Okay, that's a little bit different, convergent evolution. Now, systematists are going to use homology, not analogy, when making our phylogenetic trees. Convergent evolution is when you have similar environmental pressures and natural selection that produce similar or analogous adaptations in organisms, but they come from completely different evolutionary lineages. So what that means is convergent evolution, you end up getting the same structure or the same function, if you will, but they went about different ways to get there. Okay, so they'll converge in terms of having that same function, but there is no recent common ancestor that ties those structures together. A good example here is the Australian mole versus the African golden mole. So both of these organisms live under the ground. We notice here lack of eyes, very reduced eyes. So organisms look very similar to one another, yet this is due to convergent evolution and analogous structures, not homologous structures. Homology can be distinguished in several different ways. We're not going to get into this. Some of it is by nucleotide sequence. Some will be looking at the fossil record and looking at where these organisms are found. Just keep in mind, the more similar the two complex structures are, then the more likely that they're going to be homologous. Systematists use computer programs and mathematical tools when analyzing comparable DNA segments from different organisms. So as we found out in lab, a DNA sequence is incredibly long. So in order to be able to compare every single nucleotide, then you're going to have to use some sort of computer program to help you do that. And there's many different ones that are available, and they're actually free to use. The U.S. government has a database website that has those tools available. Mathematical tools are also going to help identify molecular homoplasies. Okay, so this is also looking at analogies instead of our homologies. So you're looking at similarities that arise by coincidence instead of by that common ancestor. Once these homologous characters have been identified, you can use them to help infer a phylogeny. So inferring, you're really making a guess on how you think those trees should be put together. Cladistics is a type of phylogeny, and cladistics is going to group organisms by common ancestry. Groups the organisms by common ancestry. We're going to use cladistics in lab, and we're going to use cladistics in later chapters as well. Definition of a clade is similar to that of a taxon. This is a group of species that includes an ancestral species and all of its descendants. So taxon was looking at each level of that hierarchy that we went over, our kingdom, phylum, class, okay? Clade, you're still looking at very specific levels, but you're looking at a species and you're tracing it back, if you will, to that common ancestor. Okay, so to be a definite clade, you have to have that common ancestor. Clades can be nested in larger clades, and we'll see how this occurs, but not all groupings of organisms actually qualify as clades, and I want you guys to be aware of this. So a valid clade is considered to be monophyletic. Monophyletic. So that means it consists of all of the ancestor species. Well, it consists of the ancestor species, so that common ancestor, and then all of its descendants. Okay, everything that came from that common ancestor. That's a valid clade. Monophyletic, mono meaning one common ancestor. Paraphyletic is when you do get that ancestral species, that common ancestor, but it doesn't include all of the descendants. You're just including some of the descendants. Polyphyletic is a tough one because polyphyletic, you're including a whole lot of species, but you're not including their most recent common ancestor. So poly means many. In that case, you're looking at the possibility of there being more than one common ancestor that may have contributed to that grouping. Now, both paraphyletic and polyphyletic groupings, neither of those are considered to be valid clades. So let's take a look at a figure here. So this is in your book. Make sure that you spend time with this. Spend time with this particular figure. Here's a monophyletic clade. Okay, that number 1 is showing you the common ancestor for this particular group 1 clade. This is including species A, B, and C. Okay. Here's group 2. This one is a paraphyletic grouping. So here, number 2 is our common ancestor. It is included in the grouping, but we're not including all of the descendants. We're including D, E, and F, but here we're leaving out G. Okay, so only monophyletic, remember, leads to a true clade. Polyphyletic, the common ancestor is here, number 3. Here's our grouping, group 3, and group 3 is not including that common ancestor. It's including this common ancestor right here for B and C. It is not including even the common ancestor for A, B, and C. And it's definitely not including the common ancestor for A, B, C, and D. You'd have to go back even further to take a look at this common ancestor. Okay, so A, B, C, and D all arise from different points. There's one here. And you're not including a common ancestor. So polyphyletic is definitely not considered to be a clade. Polyphyletic groups cause problems. So anytime those polyphyletic groups show up, then they're tried to, the systematists try to reclassify the organisms if there is a suggestion that they're showing up to be polyphyletic. It's that much of a problem. Okay, so here in practicality, this kind of shows you what's going on. We have a paraphyletic group here. Here's our common ancestor. So these are the even-toed ungulates. And of these even-toed ungulates, paraphyletic group, remember we're including some descendants, but not all. We're including the other even-toed ungulates and the hippopotamus, but we're not including the cetaceans. Okay, so this right here would be a monophyletic group. What is being shown to you in blue is a paraphyletic group. A polyphyletic group would be shown here in yellow. Now, these two groupings look very similar to one another, the cetaceans and the seals, but they are not very closely related to one another. Okay, so here we don't have a recent common ancestor being shown. You're looking at them based upon morphological characteristics, and in some cases that just doesn't work. Okay, you have to go back and look at that common ancestor. So these guys would be considered to be a polyphyletic grouping. Shared ancestral and shared derived characters are also used in comparison with that common ancestor. A shared ancestral character is one that originated in an ancestor of that taxon. Okay, so ancestral character goes with the ancestor. Then you have something called shared derived characters. Now, shared derived characters are going to arise since species have arisen after that ancestral character. So it says here, evolutionary novelty unique to a particular clade. When you're building these phylogenies, you look at both ancestral and derived characteristics, and sometimes a character can be both ancestral and derived depending upon which way you look at it. It's not always simple. So when you use evolutionary relationships to try to figure out what's going on, and you're trying to figure out who's connected with who, it's very useful to know when a shared derived character actually first appeared in a clade. Okay, so let's take a look at this. Here we have a data table. Okay, so this is a character table. So over here it shows different characters, and up here it shows different taxa. So a 0 means it doesn't exist in that particular organism, and a 1 means that it does. Okay, so here's a vertebral column or a backbone. In our organisms, the lancelet, which is going to be considered an outgroup, and we'll define that here in a few minutes, does not have a vertebral column. But everything else that we will look at does. So that's what that's telling you, is that this is a derived character that is going to evolve fairly early on. Okay? Versus down here when we look at hair, the lancelet does not have hair. The lamprey does not have hair. The bass fish does not have hair. The frog, no hair. Turtle, no hair. But when we get to the leopard, hair has evolved by the time we get to this species. Okay? So when looking at our evolutionary relationships, if you will, here's our common ancestor. Here's our lancelet, which is our outgroup. It doesn't share any derived characters with anything else. Here's formation of that vertebral column that everybody has, except the lancelet. Here's formation of hen's jaws, which everybody has except the lamprey and the lancelet. Formation of four limbs, so tetrapods, frogs, turtles, leopards, yes. Fish, lamprey, lancelet, no. Formation of an amnion, which is going to protect the embryo, is present in a turtle and a leopard. Frogs do not have this. Fish do not have this. Lamprey eels do not have this, and the lancelets do not either. So remember, we said our last characteristic to be derived was actually hair. So the only one that ends up having hair is going to be the leopard. Nobody else has that shared derived characteristic. An outgroup, I told you guys we would define this. It's a species or a group of species that is closely related to the in-group, okay, the various species being studied. But the outgroup, and I'll highlight this, the outgroup actually diverged before the in-group. Okay, the outgroup diverged before the in-group. Okay, so systematists can use these in-group species to compare to the outgroup, if you will, to figure out the differences between what are derived characteristics and what are going to end up being those shared ancestral characteristics. In some trees, the length of a branch can reflect the number of genetic changes that have taken place in a particular DNA sequence in that lineage. Okay, so here's a long length, so that means a whole lot of changes have taken place in whatever gene that they're looking at. The mouse and the human had very few changes to get whatever they needed to get for that particular gene. Varying different lengths here for our lancelet, zebrafish, and our frog. But in other trees, branch length can be different. It can be proportional to time. It can be determined from the fossil record. Okay, so here we're looking at time, again coming from that fossil record. So the length of our lines are a little bit different because now we're representing who evolved first in terms of evolutionary time. And mouse and human here are pretty equal with one another. Now, systematists can never be sure of finding the best tree. Okay, so if you've got a really big data set, then you're kind of guessing how that data set should be put together in order to get the best tree. One of the ways that it's done is by doing maximum parsimony and maximum likelihood. Now, maximum parsimony assumes that the tree that is best requires the fewest evolutionary events. So maximum parsimony, the tree that is best requires the fewest evolutionary events. So in parentheses here, the fewest appearances, if you will, of shared derived characters. Maximum likelihood is based on probability rules. And this is tied in with how DNA is going to change over time. So they look at the most likely way to get a sequence of evolutionary events. Now, which one's correct? Maximum parsimony or maximum likelihood? And the answer is both. So you use a computer program to help you build these trees, and it's going to give you different projections on what that tree could be based upon the data that you have. Okay, so there's not necessarily one tree that's going to end up being the best overall. So kind of keep that in mind. It's something that could change quite often. The best hypothesis for making that phylogenetic tree is a tree that fits the most data. So morphological, what it looks like, molecular, what the genetic makeup is, and a tree based upon the fossil evidence as well. Keep in mind as we go through this, we've talked about genetic data. Now, organism's evolutionary history is actually documented in its genome. Okay, so I know really talking about DNA is a Bio 6 topic, but talking about genomes and evolutionary history is a Bio 7. So very often when making these comparisons to build a phylogeny, we've got to compare the nucleic acids, okay, our DNA or our RNA, or even our proteins, if you will. So we're looking for similarities, and the more similarities they are, then the more related the organisms happen to be. DNA that codes for ribosomal RNA actually changes relatively slowly. Okay, so the rRNA that you see there, right here, that stands for ribosomal RNA. So this is the RNA that makes ribosomes. So the DNA that codes to make those ribosomes actually changes very, very little. Okay, so over evolutionary time, we're not seeing a whole lot of change. Mitochondrial DNA, so that MT there is referring to mitochondrial, this actually changes pretty rapidly. Okay, so looking at mitochondrial DNA can help look at recent evolutionary events that have occurred. So changes that have occurred fairly recently. Gene duplication is also another way to look at evolutionary change. So gene duplication is exactly what it says. You take a gene and you make a copy of it. You duplicate it. And this can happen sometimes. So you have a DNA sequence, and that DNA sequence actually makes a whole copy of itself and inserts that copy someplace else. So by having that more than one copy of a gene, now you can possibly accumulate changes within those gene duplications. And that can lead to a new gene, if you will. So like homologous genes, similarities, duplicated genes can actually be traced back to a common ancestor, because you can count how many times that gene has been duplicated, and over time you can figure out when it happened. Orthologous genes are found in a single copy in the genome and are homologous between species. Okay, so orthologous genes and homologous genes are very similar in definition. Okay, but here's the key. Orthologous genes are going to diverge only after speciation occurs. Okay, can diverge only after speciation occurs. So let's take a look at this. Here's an ancestral gene, and over time, this ancestral species actually diverged into two new species. Okay, so here's species A, and here's species B. Okay, so this gene accumulated a mutation here. This gene, same gene, right? But the mutation that's present in this species is a little bit different. Okay, so these are considered to be orthologous genes. So they're the same gene, but slightly different, and those differences occurred after species A and species B came from this ancestral species. So this is after speciation has occurred. Paralogous genes ties in with those gene duplications that we were talking about. So these are found sometimes in more than one copy within the genome, and they can also diverge with the species that carry them. And like I said, sometimes even get new functions. So with the paralogous genes, we can see, here's our species, and this is still the same species, okay, still species C, but after many generations, we end up getting our duplication event. And over time, we can end up getting a mutation here in one sequence and a mutation here in the other sequence. So now, for all practical purposes, these genes are similar yet different because of those changes. So this is what we term as paralogous. Orthologous genes are very widespread, and they extend across many different species. Okay, so a lot of times in searching for homologous genes, you actually will track orthologous genes instead. So one of the examples that they give is the fact that mice and humans actually diverged into two species about 65 million years ago, but 99% of our genes are actually orthologous. Okay, so they started off as being homologous, and changes have occurred since that speciation event. Gene number and complexity are not strongly linked, so don't think that an organism that has more genes is going to be more complex, because that's just not true. Humans have only 4 times as many genes as yeast, which is a single-celled eukaryote. So we would think as humans we would have millions times more, but that's just not the case. Molecular clocks help us track evolutionary time. I'm just going to mention evolutionary or molecular clocks because they're pretty darn complex, and there's different aspects that they look at to help keep those clocks on time, so to speak. So the idea here is to get those phylogenies from that fossil record, and to look at it correctly, you've got to make assumptions, if you will, with how molecular change actually occurs over time, and it can be over that long period of time. So a molecular clock uses the constant rates of evolution. Okay, we talked about evolution being constant or gradual change. That was one of the ideas that were gotten from the geologist. So that constant rate of evolution in some genes to estimate the absolute time of evolutionary change. Now, not all genes are going to evolve at the exact same rates. Remember, some genes are going to help the organism become more fit. Some genes are not going to increase fitness, and some genes are just going to be neutral, not going to cause any change whatsoever. So there's different things that can be looked at to estimate this molecular clock. Now, in orthologous genes, you can look at when the nucleotide substitutions are in proportion to the time since they last shared a common ancestor. In paralogous genes, you're looking at nucleotide substitutions since the genes became duplicated. So you're looking at different events at different times is what this is kind of bringing you into. Molecular clocks are calibrated, if you will, against branches whose dates are actually known in the fossil record. So you have to have something to compare it to get the correct time. But like I said before, individual genes are going to vary in how well they stick to that clock. So there's issues with molecular clocks. They do not necessarily run as smoothly as expected if all mutations are considered to be neutral. Natural selection, like I said, are going to favor some DNA changes because of fitness levels. And even looking at the fossil record can cause issues as well. Okay, so molecular clocks is out there. It's being used to help build these evolutionary trees, but it's not necessarily 100% accurate. So let's talk a little bit about our branch of life. Okay, so the information that we have on DNA and DNA sequences, tying it back to our fossil record, has really given us things to think about in how we classify our organisms. In early days, taxonomists classified all species as either plant or animal. Okay, so something that moved versus something that didn't move. Plants or animals, so two kingdoms, that's it. A little bit later on, and this was around actually for a fairly long period, 5 kingdoms were recognized, and you might have learned this in high school. Monera, which were our prokaryotes, Protista, Plantae, Fungi, and Animalia. Okay, but not all of our organisms fit very well in these 5 kingdoms. So more recently, within the last 20 years, the 3-domain system was introduced. So this is our bacteria, archaea, and eukarya. And the 3-domain system is what we're going to be using in class. Now, this is supported by data that comes from looking at all of these DNA sequences and comparing those DNA sequences. Okay? This gives you an idea of our 3 domains and an idea of the clades that fall into those domains. And we're going to talk about these a little bit at a time. Okay? Now, the tree of life suggests that eukaryotes and archaea are more closely related to each other than to bacteria. So let's jump back real quick. Okay, so here's our archaea. Here's our eukarya. When we look back, here's our branch point. So the suggestion is that our domain bacteria is kind of our outlier, if you will. So here's our common ancestor for life, and then our branching included our eukarya and archaea, and then a separate branch is going to support our domain bacteria. It has been documented that there's been a lot of interchanges of genes between organisms in different domains. Okay, so exchange of genes, if you will. Horizontal gene transfer is the movement of genes from one genome to another. Okay, this can be between closely related species, or it can be between species that are not closely related. Okay, so keep that in mind. Some biologists think that this horizontal gene transfer was very, very, very common, especially during the early history of life, because everything was kind of jumbled together in that ocean before the movement onto land. So instead of having a nice tree, let me go back real quick, as what we see here, the idea of the tree of life is actually this. Crazy. So domain bacteria here, domain archaea, domain eukarya, and we've got mixtures running in between each of these, especially down here at the base of our tree. Okay, lots of intermingling and a lot of horizontal gene transfer. So hopefully throughout this chapter you've realized that making phylogenetic trees are just not easy. So a lot of it's speculation based upon the data that we have, but you have to realize that we increase our data all the time. So that evidence changes constantly. I hope you've enjoyed the lecture.